Good morning, everyone, and welcome to the 2025 CRB webinar series. Today's webinar will discuss brown garden snail IPM and provide an update on Argentine ant research. I am Caitlin Stan with the Citrus Research Board, and I am joined by Joey Mayorkin, who will be introducing our speakers. Please note that this webinar is geared towards growers and pest management professionals, and the pest management methods presented, especially the pesticides, should not be followed without a clear understanding of their legal use.
Thank you, Caitlin. Please welcome our first speaker, University of California, Riverside, Biological Control Specialist and Principal Investigator, Dr. Mark Hoddle. Dr. Hoddle has headed research in his laboratory since 1997 and is primarily involved in the identification of pest problems where biological control could be a successful approach. Today, Dr. Hoddle will discuss the biology and management of brown garden snails in citrus orchards. Mark, please go ahead and share your screen to begin the presentation.
Okay, right. Thank you, Joey. Good morning, everyone. Okay, are we good now, everybody? How's that, Joey? Caitlin, does that look fine?
That looks perfect, Mark.
Okay, great. Okay. All right. Thank you, everybody. Sorry for the few seconds there of technical difficulty. Yeah, I've been asked to talk about biology and management of brown garden snail and citrus orchards. And for me, this is a return to work that I used to do about, I hate to say this, almost 30 years ago when I was evaluating various metaldehyde, iron phosphate, and believe it or not, carbamate baits for controlling snails in orchards. So we're going to be going over the brown garden snail, and we're going to be looking at different control options that may be useful in an IPM program for the significant citrus pest. But before we jump into the management tactics, I'd like to go over some of the biology, behavior, and I guess global distribution of this mollusk, because it's really quite interesting. So the scientific name for the brown garden snail is now Cornuaspersum. It's a air-breathing snail, and it actually has a lung situated within the mantle underneath the shell. And it's in the family Helicidae, which means it has a coiled shell, kind of like a turban, that sits on the foot of the slug. Many of you may know the name of the snail as Helix aspersa, and it had this name for just over 200 years before some recent taxonomic revisions moved it into a different genus. So the native range, this was very interesting for me because a lot has changed since I first worked on this snail, and some molecular analyses, which have been very well done, indicate that this snail is actually native to the northern parts of Africa, and that there are two lineages, one that's found in the eastern side of the north part of Africa, up around Tunisia. That form is actually quite rare, especially globally, after the snail became an invasive pest. And the more common form originates from the western part of North Africa, around Morocco, and from that area it spread into Mediterranean Europe. And this is the very common form of the the brown garden snail that we are dealing with here, basically all around the world where it's become invasive. So this west lineage from the northern part of Africa has successfully invaded Australia, New Zealand. It's in parts of Central and South America. It's even made it into Central Europe, got into South Africa, and of course it's here in California and other parts of North America. And some studies suggest that this snail has been moved around by humans for more than 8,000 years, primarily as a food source. So its introduction into California, it appears that it was deliberately introduced from France sometime around 1850 into vineyards along the Guadalupe River up near San Jose and Santa Clara County. By 1850 or so, specimens were then collected from around Santa Barbara. Then it was deliberately introduced into the LA and San Francisco areas, possibly for food or as a scientific curiosity. Believe it or not, some people like keeping these snails as pets. And it's been a recognized pest of citrus in California since around 1900. Interestingly, California has acted as an invasion bridgehead for snail invasions to the rest of the world. And this is probably due to our large agricultural industry, our horticulture, and the ornamentals industry, which ships live plants. And a University of Oregon analysis has suggested that the snail costs California citrus growers around $7 to $10 million per annum in management costs. So let's have a look at the invasion history here of the snail. You can see that, just a minute, please. Can you leave? Thanks. Sorry, I got people coming into my office. Okay, I got rid of them. So its native area is this northern part of Africa where the west form crept up into the southern part of the Mediterranean. And then there've been two dispersals of western lineage snails. The first major one was into California, and then the second invasion made its way down here into New Zealand. Now, what's really interesting about this is that California has pretty much seeded the rest of the world with the haplotype that invaded or was deliberately introduced into California back in the 1850s. And this has largely been accidental and mainly moved through potted plants in which the snails were either living as a shelled form or possibly as eggs that were buried in the soil of those potted plants. So all the invasive snail populations are of the western lineage, with strong representation of snails originating from France and Spain, and of course California then, because of that large population that we've had here in California for quite a while, subsequently redistributed those snails into new areas. Okay, so snail anatomy 101, these are strange looking animals. So they have a calcareous shell, 4 to 5 right-handed whorls. Very rarely you might find snail shells that have left-handed orientation to those whorls. And those shells tend to be about 1 to 1.5 inches wide and high. They have a large muscular foot, and from this foot is released mucus. This mucus is kind of sticky, and it helps to stick the foot to the substrate along which the snail is moving, and this provides the resistance to propel the snail forward. So as those muscles contract and you get those peristaltic waves moving through the foot of the snail, that sticky mucus helps move it forward rather than it sliding backwards. You're probably all familiar with the tentacles on the head region of the snail. There's 4 of these. The top 2 tentacles have photoreceptors and olfactory capabilities. The photoreceptors are pretty basic. Most scientists think that these light sensors that you see as little pinprick black dots at the end of the tentacle tend to be mainly able to distribute between light and dark and maybe shadows. The bottom 2 tentacles just above the mouth region are used primarily for touching and tasting things. And the mouth contains a strange tongue that's called a radula. And this radula has teeth on it, and that radula is attached to a large structure called the odontophore, which is moved by muscles. And as that radula is moved backwards and forwards, the food is basically shaved, and then mucus helps pull those food shavings into the buccal cavity, and then they move down into the esophagus. And you can see the teeth here on the radula. So as I mentioned earlier, this is an air-breathing snail. It has a lung, and it has a hole underneath the shell called the pneumostome, and it's through this hole that air passes into the lung. So it's very easy to see. You can just pick the snail up, roll it over, and you'll see that large hole there in the mantle through which the air moves into the lung. So life cycle. This is fascinating, and if we've got time at the end of the presentation, I'd like to, show you a very cool video of snails mating. So they're hermaphrodites. They have male and female organs, reproductive organs within their bodies, but they very seldom self-fertilize. And outcrossing is like super common amongst these snails because it promotes genetic diversity, genetic variance, which probably is useful for adaptation and survival in new habitats. And you may have heard of these love darts that these snails fire into each other. These are calcite spikes, and they contain a hormone that helps the recipient use the sperm that's receiving from the other snail, and this minimizes the chances of self-fertilization. And this is really important if you consider the fact that the sperm may be viable in these recipient snails for up to 4 years. You don't want competing snails or even sperm from within the same snail outcompeting the donor's sperm. So these eggs are laid in nests or under, say, bits of wood or maybe in the cracks of bark because they're often protected in some way. Females may lay batches of 50 to 150 eggs in a crevice in the soil or a depression, which may then be covered over. They can lay about 6 batches over their lifetime, about 700 eggs per year. And it takes about 10 to 28 days for those eggs to hatch, depending on the temperature and the relative humidity of the soil. Those young snails, when they hatch out, take about 2 years to mature in Southern California. And then those adult snails are actually quite long-lived. Some estimates indicate they may be able to survive in these orchards for around 2 to 3 years. Okay, so let's go over some basic snail biology, the 101 types of facts. As you're probably aware, the body is soft and slimy. It produces a lot of mucus and it's prone to damage and drying out. So when threatened, these snails will release a lot of foaming mucus, and you may have noticed this when you pick them up and you start tapping them, they'll retreat into their shells, and then this yellowish, greenish type, it almost looks like bubbling snot that sort of comes out around the opening to the shell. So brown garden snail lacks what we call an operculum, which is a protective shell which sits on the foot of the snail. And an operculum, as you can see in this photograph, covers the entrance or the aperture to the snail shell when the foot is retracted, and this provides protection either from desiccation, predators possibly trying to attack the fleshy foot of the snail. But brown garden snail lacks an operculum. So during hot or cold weather, the shell opening is closed with dry mucus, and this is referred to as the epiphragm. And snails, as you know, to try and reduce the risks of desiccation and drying out, are really mainly active at night when it is warm and it's also wet. And periods of inactivity or quiescence can happen during two major parts of the year. During the summer when it's hot, and we refer to this period of inactivity as estivation, and cold periods over winter when the snails shut down and they form the epiphragm, and we refer to this as overwintering. Okay, so brown garden snail damage in California's citrus. So I think it's reasonable to say that during drought years, snails may be of relatively little significance in comparison to years when there's been heavy rainfall. But of course, irrigation practices provide a lot of water in these orchards, which the snails really need to survive. When there's low levels of irrigation or no free water available, snail populations don't do so well. Moisture is really important to them because they're obviously soft-skinned and they need that moisture to keep them alive. So during high rainfall years, especially when we have these warm spring rains, snail outbreaks can occur. And some of the literature I was reading in preparation for this presentation indicated that over 1,000 snails have sometimes been counted on individual citrus trees. Up to 40 to 90% fruit damage can occur if snails can access the canopy and access the fruit. So the snails tend to feed on the outer colored layer of the citrus peel, also known as the zest or the flavado. And the damage then exposes the white underskin, the albedo, which lies underneath the colored rind. And obviously, when heavily damaged fruit is moved through the packing house, it is culled. However, it can be quite difficult to detect very small holes that snails make, and this can increase the risk of penicillin infections post-harvest. So young trees, around 1 to 2 years of age, apparently are at significant risk of not only foliar damage, which can result in defoliation from snail feeding, but the snails will also strip the bark off those young trees, which can cause mortality. Now, interestingly, one author suggested that one of the reasons why snails have possibly become more problematic in orchards, especially citrus orchards, has been through the increased use of pre-emergent herbicides and no-till weed control. Because the soil is no longer being damaged either to control these weeds, eggs that are laid in the ground, or even the adult snails themselves, they're not being killed through the mechanical disturbances that would occur if you were doing tilling for controlling weeds. And I thought this was quite interesting, an unintended consequence of advancing technologies for controlling weeds in citrus orchards. Okay, let's get into management now. And this is something that's really quite striking when you go into some orchards to see massive numbers of brown garden snails estivating on these tree trunks, drop fruit just covered in snails where they're feeding, obviously for food to get the moisture that they need, and large numbers of them just crawling over the detritus underneath these trees. So when numbers are high, you may see this type of hole formation on leaves, which has been caused by snails, possibly slugs as well, coming out to feed on that foliage at night. And sometimes you may even find the culprit sleeping on the leaf during the day, and you can see the leaf damage around it. And of course, they get into the fruit. You can see that they've eaten away the rind in this photograph, exposing the white albedo beneath the rind itself. Okay, so snail control. There's going to be a lot of information on this slide, and it basically snail control falls into really just maybe two broad categories, and at a stretch, possibly three. Chemical control, cultural control, and maybe biological control with decollete snails, which I'm going to get into towards the end of this presentation. Okay, so chemical control, many of you are probably familiar with the baits that get sprinkled onto the soil. These contain an active ingredient that's either metaldehyde or these iron chelates. And basically, you are poisoning the snail by delivering to them a poison that they eat. So for example, some of these baits may contain a bran. They may be bran-based baits that the snails find attractive to eat, and through consuming those pellets that have been thrown on the ground, they ingest a lethal dose of poison. So some of the recommendations for applying these baits to the ground is to apply them when the snails are active, possibly after a rain event when there's high levels of fog, or maybe even after irrigation. Placement of the bait can be quite important. If the bait is placed underneath the canopy but close to the irrigation lines, there's some evidence to suggest that snails may be able to recover after they've eaten a dose of that metaldehyde, for example, because they're in a moist area, they're getting the water that they need. So one of the recommendations is to move the bait applications out to the canopy margin, which moves the snails away from that moist zone underneath the tree canopy. And when they get a lethal dose of metaldehyde, it's harder for them to move back into that safe zone of shade and moisture, which may facilitate recovery from a heavy dose. Some applicators may even put the baits out between the rows, which really pulls the snails out into an area where after they've consumed the poison, it's going to be very hard for them to get back to an area where they can get the moisture and shade that they need if they're going to, which may help them recover from a dose of the toxicant. Some work suggests that smaller pellets offer better efficacy than larger sized pellets, probably because the snails are better able to consume them. And Ted Fisher, an entomologist here at UC Riverside, who's done a lot of work on snails, had an interesting CalAg article that he published in 1975, suggesting that it may be possible for snails to develop resistance to metaldehyde, and that this in the past has been a problem Caltrans has been dealing with when trying to manage snails along the roadside plantings that they're responsible for managing. If anybody has any comments on this, I'd be very interested in knowing about that. So there are these molluscicides, and I've chosen one example here, methiocarb, mesurol. It's not available in California anymore, but it illustrates a different mode of action. So these insecticides that have been reformulated for the control of snails affect the nervous system of the snails. It puts them into a type of paralysis, similar to what happens to an insect or even to a human if they get an overdose or an exposure event to these insecticides. The metaldehyde and in particular the iron chelates affect the stomach function of the snail, so they ended up basically dying from starvation rather than having their nervous system interfered with. And of course, with all of these products, and as Caitlin mentioned earlier, you need to follow the manufacturer's recommendations and state level regulations on applying these products. So there are other products that have been looked at for controlling snails. For example, Dazotol is a combination of these capsaicin and garlic and onion extracts that can be combined in a liquid formulation that is then applied to the soil as a drench, or maybe even applied to foliage. There's even some work that's been done that suggests that clove oil may be quite effective for controlling snails that are using the soil for either crawling over it or even laying their eggs within it. But this is really only useful, I think, if you're trying to control these pests in a nursery where these plants are potted up. So cultural control has received a lot of attention, and one of the recommendations is just try to keep your pest snails out of new plantings. Make sure that you're not introducing eggs, adult, or maybe even young snails in new citrus plants that are being planted out. Trunk barriers are sort of a technique that have been around for quite a long time, especially these copper-based trunk barriers, and I'm just going to get into that a bit later. But recently there's been the development of plastic shields, which have been pioneered by our citrus growers up in the Ventura area. So with these barriers, you apply them to the scaffold, you apply them to the trunk below the scaffold branches, and it's recommended, at least for the paints or the copper barriers that go onto the trunk, they need to be at least 12 inches wide, and make sure they're up on the trunk out of the sprinkler zone so they don't get washed off prematurely. Again, Ted Fisher at UC Riverside, who worked on this way back in the day, indicated, at least from his limited field trials, that these barriers may be about 90%, 7% effective in keeping slugs and snails from getting up into the canopy. Skirt pruning is recommended to get the skirt or the citrus branches and leaves up off the ground. So this makes it more difficult for snails that are moving along the ground to get onto a branch and then work their way up into the canopy where they can damage fruit or leaves. So the UC IPM recommendations here, it's about a 24 to 30 inch space to lift the branches, the canopy up off the ground by that distance, and then combine trunk skirting with the use of a copper spray, and I'll get into this a little bit, a couple of slides, either putting some sort of metal barrier around the trunk or painting on a Bordeaux slurry, which is a mixture of copper sulfate and lime. Also, weed and debris management under trees is important. This removes favorable habitat for snails, especially shelter where they can get out of the heat or cold and avoid direct sunlight and maintain their cells in areas that may have a more favorable humidity for them. Basically desiccation avoidance habitat. You want to remove that for these snails. And biological control, well, there's a lot of discussion around whether or not decolate snails can attack and successfully kill brown garden snails and provide population level suppression of these brown garden snails. And we're going to look at that towards the end of this presentation. Okay, so here's one of the plastic barriers that's been developed, the snail jail. It's an interesting video to watch. So it's a plastic barrier that goes around the trunk, and you can see it's got all these spikes around the outer edges, which prevent the snails from moving over those barriers to get over the top of the barrier, so then they can access the trunk. And basically these barriers are put around the trunks of the trees, and then the snails work their way up, but they can't cross the barrier because of those spikes. And then if they're sealed well enough, there should be no gap between the inner margin of that barrier and the trunk itself. So something that's come out of South Africa recently is the bait chain. This is basically a cord that you tie around the tree, and attached to that cord metaldehyde pellets. And the snails move their way up to this cord. They don't seem to cross it readily. They probably feed on the metaldehyde, and then that's it, they're done. And field trials in South Africa, at least in the apple orchards, suggest that these bait chain wraps around the trunks may be as effective as controlling brown garden snail as broadcast pellet applications to the ground. I'm not aware of any work being done in California on this, but it's interesting. But I imagine it must be very labor intensive crawling. Well, at least citrus, you'd have to crawl on the ground to get underneath the canopy to apply these bait chains. So skirt pruning. So I was reading about this, and I found this was quite interesting because I didn't really know much about it. But one of the authors on this article indicated that, if you're pruning older trees for the first time, you will get fruit loss, and this has been a concern with growers. However, the argument made for even pruning older trees is that fruit to the ground may be damaged or of poor quality, and they're likely to be culled if that's the case. And furthermore, once the skirt is lifted up, trees tend to compensate for the loss of fruit lower down by increasing fruit production higher up in the canopy after the skirting has been completed. The other benefit is you can get good Argentine ant and fuller rose weevil control through lifting the skirt up off the ground, and reduced instances of Phytophthora brown rot and gummosis as well. Other benefits include easier inspection of the irrigation systems, because you don't have a lot of foliage close to the ground, which impairs your ability to look at the sprinklers or the micro emitters underneath the trees. And also, I thought this was quite interesting, that lifting the skirt improves frost protection because you can get better movement of cold air through the canopy and underneath the trees rather than it being stagnant, which would likely decrease the temperatures to a level which may end up damaging the trees. Okay, so these copper barriers, there's two ways to apply these copper barriers to the trunks of citrus trees. Trunks can be painted or sprayed with a copper Bordeaux mixture, which is a mixture, I think, of lime and copper sulfate. UC IPM recommends that these bands should be at least 6 inches in height up the trunk, and obviously they need to be below the first scaffolded branches and out of the irrigation zone. So they can be prone to washing off, and longevity may be increased if the Bordeaux mixture is mixed with white latex house paint or possibly even linseed oil, which may increase field longevity. Permanent copper barriers, if you want to consider them, think of them in that way, can consist of copper foil, copper mesh, and I've seen one video which was quite interesting. The guy shown down here got, large rolls of copper wire, thick grade copper wire, and stripped the insulation off it, and then just basically wrapped the copper wire in a thick band around the tree trunk. And you can see that the snails piled up here and just wouldn't cross the copper barrier. But these permanent barriers, copper barriers that you apply to the trunks, need to be set up in such a way that as the tree trunk grows, the band itself can expand itself. And one way to do this is to nail or attach permanently part of the band, and then where the band pieces overlap, to have a couple of paper clips that hold them snug. But then as the tree grows, that band tends to expand with the increasing girth of the trunk. Longevity is probably greater than 5 years in the field. I don't think I have ever seen copper bands applied to large numbers of citrus trees in commercial situations. And if there are examples of this, I'd like to know about them, because I'd be very interested in going out and having a look and getting some photographs for these types of presentations. So nobody seems to really know how these copper bands work, but they are obviously very effective at deterring snail movement up and down tree trunks. And it's been proposed that maybe there's something to do with the copper, and that with electrons moving through the copper, different gradients between an anode and a diode, a cathode, that you end up creating a shock to the snail's foot, and they don't like that sensation when they touch it. Okay, the other thing that's really interesting about these snails is that they seem to aggregate, and this has been put down to the snails having really good homing behavior. And then once they home in on a like a resting site, they tend to show high levels of fidelity to that site, and they will return to it time and time again. And experiments in the lab, at least, indicate that brown garden snail will follow the mucus trails of individuals within the same colony cluster. They have a high preference for following mucus trails of neighboring snails that use the same aggregation site. They will follow the mucus trails of snails that aren't within the colonial colony within which the snail is most accustomed to, but they will also follow those snail trails to other resting sites. But they prefer to follow the trails of snails with whom they tend to aggregate together on the trunks of trees, for example. Interestingly, experiments have shown that if you move the snails around, they'll find those trails again, and they will move their way back to the tree trunk that they were removed from. So they seem to have a home range, and they forage within that home range, and they tend to estivate and overwinter within that home range. And as you can imagine, that home range is not very big, maybe a few meters. So there's been some speculation as to, well, why do these snails aggregate? Well, in some species, not brown garden snail, but it's thought that the aggregations may protect against, I should have dehydration there, not hydration, and maybe even predation, safety in numbers. So homing to these microhabitats, it's been speculated for brown garden snail, may favor survival in marginal areas. If you get too far away from these irrigated zones, you may not find a good place to get out of the heat, or if it's too cold, and you'll be subjected to mortality. But then other researchers have postulated that maybe homing in these snails is really not that strong after all. And there's just a limited number of aggregation sites for these snails, so they just all end up piling up together in the same areas when they follow each other's mucus trails. Interesting ideas, which may be, may be valuable to explore as possible control strategies in the future. Right, let's turn our attention now to decollete snails. There's a lot of discussion about how effective these facultative predatory snails are for providing brown garden snail control in citrus orchards. Okay, so these snails are native to the Mediterranean basin. Not surprisingly, they've originated from parts of North Africa, Spain, France, and Italy. Similar native range as to the brown garden snail. And the first record of the decollets was in the United States back in South Carolina in 1813. They're hermaphroditic, just like brown garden snail. So both snails have the gonads of both sexes, male and female gonads in a single individual. They're facultative predators. They are not obligate predators. This means that they may occasionally feed on meat, another snail, for example, if they can get it, or if there is plant food around, they may even prefer to eat the plant food in preference to hunting down a snail, attacking it, and feeding on it. It's not a plant pest, but it will readily eat plant material that's on the ground. And I was reading about their preference for carrots. They won't eat carrots that are underground. But if you lift the carrot up, cut it up, and put it on the ground, decollets will eat it. So I thought that was quite interesting. They're not prone to climbing like the brown garden snail. They prefer living on the soil, hiding under debris that's lying on the soil, and they may actually even burrow into the soil. So this means that brown garden snails and decollets, have these different spatial preferences, which limits the opportunities for interaction with each other. Similar to brown garden snail, they flourish in well-irrigated areas and they prefer warm temperatures. The first official record in California of decollet snails was in Riverside, 12th of January 1966. Ted Fisher was sent some specimens of these snails that nobody had seen before. He went out and had a look, and yep, sure enough, he found populations of the decollet snails. And based on how widely distributed these snails were once people started looking for them and the densities that they were finding, he estimated that they may have been introduced into California at least 10 years before the official record in the state. He even speculated that maybe they were deliberately introduced, but there is no evidence for that at all. So they could easily have been moved on potted plant material, similar to, in a way similar to which brown garden snails get moved around as well. So because it has this predilection for soil, living in the soil, burying itself in the soil, hiding under plant debris, it's not surprising that this snail also has been moved around the United States. It's present in Mexico, the Caribbean, South America. It's even in England, China, and Japan. And it's the trade in live plants which has likely moved these snails around. But however, their impacts on pest snails and native snails is not really well understood. And
Dr.
this is, yep,
Not
if
to
I,
interrupt real quick, we are at time, so we
oh,
do
okay.
wrap it up.
All right. So I've talked too much, so I will stop there. Well, can I just go to one slide, please?
Absolutely.
Okay. So this is what everybody likes to see. Decollets attacking, well, in this case, being close to brown garden snail. But do they really result in successful biological control? Ted Fisher, the entomology professor at UCR, was very enthusiastic about the biocontrol potential of these decollets. If you have a lot of decollets in your orchard for more than 6 years, you end up with lower brown garden snail populations. But this observation is not consistent. So Rory McDonald and I, we reran some of Ted Fisher's bioassays, and we found results that weren't particularly compelling for the biocontrol potential of these decollet snails. They'll only eat 1 to 2 brown garden snail eggs a day. We had to dig these up out of the soil and place them on filter paper for that to happen. Probably not going to happen in nature. They'll only eat very small snails that are less than 0.5 of an inch in diameter and at very low rates, less than 4 a week. However, you give the decollet a preference between a very small snail and carrots, it prefers to eat the carrots and it won't attack the small snails. I think we just need to do some field trials to figure out what's going on. So this photo shows big snails, unlikely to be attacked, small snails, which maybe could be attacked in the field. Okay, so why do brown garden snail populations decline when their decollets introduced into the sites? I think it is a resource competition effect. Decollets are more effective at removing foods that the brown garden snail also prefers to feed on. And we see this happening in orchards. It's definitely removing a resource from brown garden snail. And if these snails, pill bugs, can do this more quickly than the brown garden snails, then the brown garden snail is going to be at a disadvantage. Okay, so some conclusions. Brown garden snail can reach incredible population densities in these citrus orchards, even when decollates are abundant. After reviewing the literature, in my opinion, the snail control is pretty stagnant. Hasn't really changed greatly over the last 50 years. You know, we use baits, skirt pruning, trunk barriers, habitat modification, possibly decollate snails. And now we've got a whole bunch of other snails that are in the citrus orchards, you know, white garden snails, milk snails, you know, what are these species and what are they doing in these orchards? And I think that for any IPM program against these snails, you're going to need to consider a combination of some of those strategies that I discussed earlier. All right, we don't have time for the video. I've talked for too long. I will leave it there. Thank you very much.
Thank you very much, Dr. Hoddle. For now, what we'll do is we will hold questions until the end of the next presentation. But I will go ahead and please help me welcome our next speaker, CRB Integrated Pest Management Entomologist, Dr. Ivan Milosavljević. In his role at CRB, Dr. Milosavljević is responsible for developing applied citrus pest research projects with industry input and also leads the California Citrus Research and Field Trials, known as California CRAFT project, which is focused on strategies for mitigating Asian citrus psyllid. Today, Dr. Milosavljević will discuss Argentine ant management innovations in citrus orchards. Ivan, please go ahead and share your screen to begin the presentation.
Thank you, Joey. Thanks for the introduction. Let me share my screen. Can you see this?
There we go.
Okay.
Yes, you're good.
Well, good morning, everyone. There was a great presentation, Mark, by Snails, and so by you regarding the snails, we have very much they have increased in numbers. So I just want to say before I start, it's a true honor to go after Mark. I have spent 7 years in Mark's lab working on different projects prior to joining the CRB. One of the projects that we have been developing over time and we're still continue to develop is the Argentine ant management. So it's kind of a shift in research topics right now, but I just wanted to highlight that it has been a privilege working in Mark's lab. So Argentine ants are also invasive like brown garden snails, right? They're not, as their name suggests, they're not native to California. They have been introduced here since 1905. That's where the first record dated in that year. As their name suggests, they're native to South America, really, to the basin of the Paraná River in Argentina, really, and some parts of Brazil as well. But they are the ant tramp species, really, the one and only that has been moved by humans throughout the world. They are pretty much present on most of the continents, and they have been moved with the soil or with plant materials, and they have established successfully pretty much everywhere they have been moved to. One of the reasons why they're so successful as an invaders is that one sentence, they're polygynous, polygamous, and genetically homogeneous, right? Which means, translated into a layman's words, that they can form these super colonies, and they are formed of multiple nests that are interconnected, multiple queens within those nests, usually below soil, and they are all kind of related. They groom each other. They don't fight with each other, just interfering with their food sources, but instead they will join forces and just outcompete and outnumber anything that tries to interfere with their food. And in that terms, in California, most of the native species have been displaced by these native ants, their food sources, and in such a way, the native ants, they can displace the bumblebees and honeybees. That has been documented as well as some larger animals like hummingbirds that try to interfere with the pollen and nectar these ants are foraging for. If they're interfering with their food, they will just outnumber these and chase them away, these birds as well as these other organisms. And this is why they have been so successful across the world. In citrus orchards, their nests are located below the soil, so below the trees in the soil, right? As I mentioned in the previous slide. And the biggest problem we have is that they have a food source that is readily available to them in the form of a honeydew. And they have formed, as we called them, these food for protection mutualism with these sap-sucking pests that we have in citrus. So we have mealybugs, we have brown soft scales, we have other scales, we have as well the Asian citrus psyllid that has recently, or not recently, over 2 decades ago, almost 2 decades ago, invaded our California. But we have these, as we call them, sap-sucking producing pests, or these honeydew producing hemipterans. they're feeding on our plants and they're the main pests. The ants are not the pests themselves, but because they provide these different services for these main pests, they're a pest of concerns to us. So these main pests, as I call them, the sap-sucking pests, will feed on a phloem sap, and some of them will transmit bacterium or viruses or whatever that will go into a plant and will eventually kill the plant. They will be damaging. They're causing the direct damage. But as they feed on this phloem sap, which is also rich, very rich in sugars, they can't process all that sugar. So they have to excrete that sugar in such a way in the form of a honeydew. And it's basically, as we call it, a sugary poop in layman's words, right? So they will come to the ants will come to these honeydew, and they will see it as a sugary food source because they're a liquid sugary feeding ants. They like sugar. They like anything that has sugar in it. And they will protect different services. They will provide different services for these pests. They will sanitize after them. They will even move them to different locations within the plant to prevent overcrowding, because essentially these hemipterans will feed more, they will reproduce more, there will be too many of them, they will not pay attention, and they will drown in their honeydew. And to prevent that, these ants will move them to a different plant, the clean parts of a plant. Well, they were provide them with a clean surface, more food, well, they can produce, they will feed more and they will produce more colonies, which in turn will produce more ants because they will need more ants and more workers to find a way and collect all that honeydew. And essentially we'll have a dual problem, right? We'll have more pest colonies as well as more ant problems in our orchards. Now, some estimates suggest that you can have about 1 million ant visits per day. They're mostly on a single tree in citrus. And they're mostly located in Southern California. They're a big problem here in Riverside as well as San Diego, throughout Southern California, also in coastal areas. But some parts of San Joaquin Valley have also documented the Argentine ant presence and problems in their orchards. Some growers are complaining on these ants, right? The problem is that the biggest problem is that they are protecting these colonies, again, from anything that tries to interfere with their food. And if we are, as a PCAs, growers, practitioners, IPM practitioners, trying to implement an IPM program for these pests in such a way that we want to incorporate, for example, the biocontrols or the biocontrol agents such as parasitoids or predators, then we'll have a problem, because these ants will protect about 90% of these colonies of any sap-sucking insects, as well as 55% of ACP, what has been suggested in previous studies. And in such a way, if some parasitoid or predator tries to interfere with their food, they will again outnumber that predator and simply killing it off or chasing it off the food source. And essentially our biocontrol efforts will not be as effective as we want them to be. So I just have a couple of slides to show. This is the mealybug infestation. It has been on our radar as one of the pests that has reemerged as a big problem throughout California. And they can have a pretty huge impact on our fruit, right? The infestations will cause a lot of sooty mold and a lot of mold to it, and you'll have non-productive fruit, non-marketable fruit, right, in the end. And pretty much everywhere you go and see the mealybugs, you'll find an ant on them, because these ants, especially all the sugary ants, whatever the problem you have in your orchard, but especially the Argentine ants, will prefer them. And that is because they are producing pretty much the liquid honeydew, which these ants really love. They will feed on this liquid, they will take it into their mouth, the workers will, and they will just move down to their nests below the ground, below every single tree, recirculate that product, that whole food that they got, and eventually they will serve as a feed to their brood and as well to their queens. And this is the way they function. The foragers will find the food, they will take the food into their mouth, they will go and feed their nest mates by trophallaxis, as we call it, or mouth by mouth is what they use. And in such a way, they will sustain the colonies. The ACP I mentioned, they also prefer, but they prefer less compared to the mealybugs. And really that's the only document that we have is that because the honeydew that these ACP nymphs are producing is not liquid, it's more filamentous. It's a little bit more solid. It's still a liquidy, but you still have a more solid component to it. And that could be probably the primary reason why they're less preferring ACP nymphs comparing to the mealybugs, for example. But yet again, if you're trying to implement the biocontrol program in urbans, in commercial orchards, you're going to have a problem there, because they're pretty much going to be still present on these nymphs wherever you have the nymph problems or ACP problems. So this is really what happens. And this is in urban areas. This is the study that we did. And on this video, you can see the small wasp that is being removed by these ants, Argentine ant workers, from these ACP nymphs. Now, this wasp is brought by Mark from Pakistan, and in an effort to establish the classical biological control for ACP in urban areas. It has been our primary tool in urban areas. It has been the one and only component that we can figure out and implement in these vast urban areas and urban backyard citrus to control the ACP nymphs. And it has been very successful. We have found a rate of about 70% decline since this program has been implemented. However, these wasps, are very expensive to produce by the state. They're being released by the states, and they're very expensive to reroute and release in these areas. They're still being released, basically. So whenever these ants were present, is what we have found, you will have a reduction of about 50% of this parasitism rate, and you will have a triple the numbers of ACP nymphs compared to those trees that didn't have the ant presence on them, which creates an issue, right? It caused an economic issue as well as an IPM practitioner's issue. If we want to establish the biocontrol agents, we're going to have a problem if these ants are a problem, if these ants are present on these trees. So the removal of the ants is essential to this program as well as to the other biocontrol and IPM programs for these sap-sucking pests for sure. This is a study that we did in Mark's lab. This is a study that has been done by a PhD student in our lab, Kelsey McCullough. I had a privilege to help with this study, really, but we wanted to see if we control these ants, is that going to be really beneficial to our biocontrol programs for multiple pests of concern? And we had a soft scale, brown soft scale, we had the citrus mealybug, we had the ACP, as well as the California red scale. Now, California red scale doesn't produce really a honeydew, but because it's just being present on trees where these ants are present, it will be also protected by these ants if something tries, a biocontrol agent, a parasitoid tries to control them. So our study was done in 6 different orchards in Southern California, in commercial orchards. And we used this baiting technique, basically a method that has been established previously. You can see them in the left far corner on this photo. The ant pros, the bait stations, the plastic bait containers that you can fill up with, you can purchase them online, really. They are commercially available. And you can fill them up with a 25% sucrose solution or sugary water, which ants love, and it will attract ants to this food source. But you can also add a little bit of a chemical, a very small amount, untraceable, of, for example, thiamethoxam is what has been proved previously to work prior to this study. And this is exactly what we used in this study, about 0.001% of thiamethoxam, which is a very, very small amount. It's like hundreds of times less than a label use of thiamethoxam in citrus orchards. And it's very effective because the ants will come to a sugar, and instead of just picking up sugar, they will pick up the poison. The thiamethoxam is really good because it's slow acting. They will pick up the thiamethoxam in their mouth, right? And they will go below the soil, and like with the honeydew, they will feed that poison to the queens and the nestmates. And essentially, when the queens are dead and all the brood is dead, you will have a colony collapse and you will have a lower numbers over time. And this is exactly what we wanted to see. Now, in this left upper graph, of charts, you can see that this was implemented every month. So we have to service them every month. And in just 3 months, we had a really good control of ants. So here in yellow line is the trees or the plots that were controlled compared to the white line that were the plots that didn't have a control in them. And after just 3 months, we had a regulation or reduction, of about 95 to 96% of ant numbers, and we kept them low throughout the study. So this is exactly what we wanted to see, right? We wanted the ant control. Now we also looked at the pest numbers over time, right? So prior to application in treated and untreated plots and after the application, in this right upper chart, you can see in the right portion of it, the treated plots, right? The treated trees had a reduction. We have seen that all pests were pretty much annihilated, and they were reduced by over 98% in pretty much on all the twigs, the fruit, and the flush, and that includes the ACP as well. They all went down by significant numbers, and this is perfect. This is what we wanted to see again. When we look closely at the parasitism rates, because there was a lot of parasitism around, we have found on pretty much all these different twigs, flush, and fruit, different components or parts of the tree, that for the mealybugs, the scales, and the California red scale as well, the parasitism was higher in these treated trees compared to control trees, which means it was working. We have provided a service and a window for these parasitoids to jump in, and they were actually doing a pretty good job in reducing these numbers of these pests on trees. But not, as you can see here down in the left corner of a slide or a left chart, that left lower chart circled in yellow, the ACP parasitism was not different between the treated and untreated trees, which makes sense, right? The Tamarixia is not being released yet in commercial zones, and it hasn't been really established in commercial areas. The parasitism that you can see, it might get from a neighboring urban releases, but it wasn't significant enough to reduce their numbers. So what was really going on? Because we have seen less ACP, much less ACP, but parasitism was not doing any good damage. And when we look at the predation rates, and this was really perfect, we have found that our native predators, like syrphid fly larvae, were pretty much ideally correlated with these ACP colonies on the flush. And they were the ones that were responsible for controlling the ACP in our plots. So that means that we have an allies in the form of our native predators in these commercial areas, and that we have to pay more attention to nurturing them and providing a better service for them so that they can kick in and do a better job for us in control ways. And this is exactly what they look like. In the left corner photo, you can see an adult syrphid fly. It's a fly, it will fly. They're hoverflies, right? So they will go above the flowers. They like nectar and pollen to feed on that, and they will stay put as long as you provide some flowering plants for them. And they will lay eggs next to their food source for the larvae, because larvae are actually predators. And this is just a species that we have here in California, in Southern California, Allobrapta. But other species are behaving in a very interesting ways. And there are other species from Europe that have a completely different behavior of controlling the pests. Very cool. I'm not going to go into a lot of details, but this is our predator. The main predator right here that we have in Southern California, for example, in citrus orchard is Allobrapta. Their larvae can feed and consume about 450 nymphs, ACP nymphs, before turning into adults. And these are the guys that we really want in our orchards. So this is just a short slide to explain everything, right? And just related to ACP. We have Tamarixia in urban areas. We have a biocontrol. We have a biocontrol option in a form of a native syrphid flies here in commercial zones. And then we have these ants that are a problem. And the only way now we know from this study, if we control these ants, this provides a proof we'll have a better services in both of these areas, and we'll have a better control, not just ACP, but other pests as well. So this is a route that we took on. And Mark and I have worked on this for quite some years right now. I think together for 5 years while I was in his lab, and we continue to do so. And it has been over 7 years now of establishing really a control program for ants, an IPM program for ants, so that we can have a better IPM program for these sap-sucking pests in our citrus orchards. And it's a bottom-up approach, right? You need to control the ants that are coming from the soil so that you have a better services in the canopy. So this is a three-pronged system, a dual active program, as I mentioned, but a three-pronged system that we have established. We have evaluated these systems individually or methods, but also we have evaluated them as a full component and a full IPM season-wide program that we have established. So I don't have time to go into cover crops. They're very beneficial. We have found that, for example, having a listum in your orchards will have a better service for these syrphid flies. It will attract more syrphid flies. It will not be attractive to ants, and you'll have a better shelter and food for these syrphid flies to kick in and to stay put in your orchards and provide a better service for you. But I'm just going to focus on these two individual components here, and that is how we streamline the process of automating the ant monitoring, which has been a problem over time in citrus, and then how to better control them. And these biodegradable hydrogel beads have been developed as a response and the request from the DPR to removal or deregistration of chlorpyrifos, which was essentially used to control these ants as a spray program. But the problem with chlorpyrifos is in just controlling the ants, the problem was that you will spray it on the surface, you will spray much larger volume compared to these beads. It will not be targeting ants, but just foraging ants that are on the ground, on tree trunks. And you will kill off the workers, but you won't have a good effect of queens and the brood that it's below the soil. And they will just repopulate the area and you will have problems all over again. So the baiting program that I explained previously with the bait stations, the Ant Pros, and the one that we have developed in these gels is a far more targeted and better approach that you will have. But I'll start with the sensors. So it's very interesting, and especially for me, I'm a little bit taller, and the traditional way is to go below every single tree trunk or a tree canopy and to envision the imaginary line on a tree trunk and count the number of ants that go up to the canopy and below to their nests as they forage the food. And you just count them over a 1 minute, 5 minutes, whatever you want to do. And that will provide the best estimate of ant numbers on your trees, coming to your trees for food, right? So they also like these irrigation lines. And if you have citrus, you have these irrigation lines, you're going to see them on these lines going between the trees, moving between the trees as well. And we have come up to a conclusion that this might be a better way of counting them. Because when I go and below every single tree, I get tired. I will stand up, I will hurt my back. It's just unfeasible over a time. And it's much better if you just focus on something that you can actually count them on. And that is exactly the irrigation lines. They're acting as super highways for these ants. They like the surface, the smooth surface that the irrigation lines provide. They're a little bit more cooler when it's warm weather, and they love water and moist environments as well. So we counted the number of ants on these lines, and we counted them on the tree trunks, and we have found that with about 85% accuracy, if you look at the numbers on lines, you will pretty much be very effective in predicting the ant colonies and numbers and densities on your tree trunks as well, which is great. So if you look at the lines, you can see the numbers that are going into that specific tree. Now, this is even a better way. You don't have to count them by yourself. We have developed, and this is still in development, this is a prototype, these sensors that can automate the counts for us. And the way they function is that you attach them to these irrigation lines, and they have these infrared beams that you can see in the video. And as the ants pass this infrared beam, it will provide the counts for us into the cloud system, right? It will just show us 1, 1, 1, 1, and you will have a binary count of these ants passing these imaginary lines on these irrigation lines. They're connected to the gateway, which is basically an antenna that you can set up on a high grounds on the antenna on a house, and they will collect the data from these individual sensors that you can distribute in different parts of the orchard that have a problem with ants. And it will transmit the data into the cloud and into the phone. And we have developed this app that you can see on the left portion of the slide. And each of these rows represent a single sensor, and each of these fields represent a 5-minute count of ants every hour. So they can provide the exact numbers for us of ants passing again these lines every single 5 minutes, every hour, every week, or every day, every week, and throughout the whole year. Which is what we want to see, right? It can provide us even with the estimates of ant numbers during night, because most of these ants will forage during the night time as well. And we didn't know that for a fact unless you go and count them during the nighttime, which is a very hard thing to do. But now with these sensors, we have found that they're pretty active throughout the nighttime as well. What you can use, again, you can use them as an IPM tool. You can see in the right upper portion, and I want to thank Sun Kwon, who is a master student in Mark's lab, for providing this data upfront before they are being published. But you can count them over the seasons every single day, right? And this is exactly what they have been used them to see a seasonality of Argentine ants in citrus orchards over a whole year. And here, the higher the numbers, the lighter the color of the blue is. And as you can tell, sometime in May and July, they will start to build up their population slowly. And then you can have the highest population over summertime. So from July to October and going into November as well. They will be active throughout the year, but the highest numbers you will have in this period. And if you want to focus your control efforts, you might want to focus them where their numbers are the highest, or go into a May or July and attack them while they're in lower numbers, as well as the pests are trying to establish as well. So maybe that timing is better if you want to control them. They're also equipped with the temperature and moisture sensors, and it's a lower right slide that you can, or a graph that you can see. You can basically tell at what time of the year and what time of the day, at what temperature they're the most active at, and you can correlate that to their numbers over a time and over a season as well. So it's a very efficient IPM tool that can be used by growers if it gets developed, fully developed, and commercially available. Gels, right? Everybody's here because of the gels. It has been a while, right? Both Mark and I have worked on this for 5 years, like I mentioned, together. We have David Haviland that is helping with some of the trials as well. He has been doing some work with some other gels, the polyacrylamide gels that I'm going to explain. So between the three of us and some other labs, it has been like 7 years, quite a few millions of dollars invested by the DPR. And finally, we got them developed, and the efficacy data is present, and the non-target effect data is present. We're now focusing on registration, right? So now it has been 7 years, and I can probably say that I'm more optimistic than before, but I'm hoping that that's the case, that the growers will have them for a use soon. I don't know when, but we're still fighting that end on the regulatory upholds, and we're trying to prevent those and then try to streamline the process of regulation. But really, there are two types of gels, and I'll just focus on the three more slides here. The first type is the biodegradable hydrogels, that, as we call them. These are the gels that Mark and I have been developing, and we have been developed these gels. They're made of food-grade materials, the sodium alginate and calcium chloride. They are used in the food production industry. And as soon as you mix them at a certain temperature, it's called the cross-linking. Essentially, you will have different forms of these gels. You can put them through a shower head, and you'll have these beads. This is what we wanted. We wanted them to be as small as possible, to cover as much area as possible, to biodegrade as fast as possible, and to be consumed by the ants as fast as possible. But kids are making them really, you can watch the YouTube videos, they're making the gummy worms, and they're really safe to be built as these small beads. When you put them into like a tub that has a sugary water and the chemical inside, they will absorb that chemical in the sugary water, and they will become a miniature bait stations. And this is really where the problem with the registration now begins, because they're essentially becoming a carrier for this insecticide in a small bait stations. So they have a same method as the bait stations I explained before at the Ant Pros. The ants will consume them, they will take a liquid out of them, and they will feed the nest mates, and the colony will collapse. But it's more targeted because and more sustainable. They will biodegrade. You don't need a servicing, and essentially you can distribute them below the trees, and they're far more cheaper than those Ant Pros that I showed. And really, we have collected a lot of efficacy data. The thimertoxam at the 0.001% again was the golden standard. It works really well. If you use the organically approved spinosad, if you use that one at the 0.01%, a little bit higher rate, it will be as effective as thimertoxam, which is still below the label rate for spinosad in citrus. And the rates that you want to use that we have found is the 5 to 10 gallons of gels per acre, and the 25% sugar has been used as the most effective attractant. Now, Mark's lab is still doing the non-target effects, and we are seeing very few non-target effects, which is a really good plus for us with these gels, and more data that we can supply to DPR and registrants for them to use and register these gels more effectively and faster. The good news is that they have been exempted from a registration as adjuvants by the DPR recently. So this means that you can make them at your farm and deploy them as gels, but when you mix them with a chemical, you can't use them. So they're still illegal as a chemical because they're off-label use, but we're making some progress one step at a time. The polyacrylamide gels that you see here on these photos are already pre-cross-linked crystals, the soil moist that you can buy from the Amazon as a crystals, as a soil moist. You can use them to collect water, right? But when you mix them with the water and sugar, they become again the carrier and they're illegal. The problem with them is that they're still pending registration because they're being produced commercially, and the producers of these gels, they don't have the registration for ag use. And this is where David Haviland is now working with the DPR and these registered to try to get them registered as much as he can in such a way that it can be used in ag systems as well. So for now, the biodegradable hydrogels have a better pathway of being registered first compared to these gels, but they're both equally effective ways. You can distribute them with these seeders below the trees, and they will have a very good effect. The same rates as for the biodegradable hydrogel beads apply. We have tried the 15% sugar last year. It didn't work, so you need at least 25% sugar. And then again, we are trying again this year. We're collecting more efficacy data on when to apply them. We're trying the spring application and to see if that will help in knocking down the ant numbers and pest numbers over time, if we can do that more targeted in a better way of controlling them while the numbers of both the pests and ants are still low and building up. And previous studies, published studies, have already shown that these gels in different systems had very low non-target effects. So this is a slide that I have compiled. I'm not going to spend too much time. I already explained this. This is a pathway that we have taken so far. You're feel free to do a print screenshots or just take a photo of it. But we are now stuck with the biodegradable hydrogels at the 24C registration for etymetoxam. That will be the first chemical to go through. We have had some regulatory, as you can tell from all the text, it has been a process to try and get them registered. We're not there yet, but the DPR has been very responsive, and we're trying to get them as soon as possible into your hands and for control of ants in citrus, which will be essentially my ultimate goal working on this for so many years, right? So other methods that Mark's lab is currently doing, and I'm a part of it briefly, is that they're looking at stopping the ants on these irrigation lines. This will be my last slide. So just some kind of a physical barrier, applying a mulch to irrigation lines, or just applying the contact chemicals, such as, for example, a fipronil that these ants can carry on their legs and bodies to their nests and poison the queens and the workers. Could become a tool as well, but the whole IPM program is being developed. We're collecting a lot of efficacy data, and we have moved forward for a long. We have made a really good progress in such a way that this is the first of a kind IPM program for managing ants, so that we can manage our pests better. So with that, I'm just going to stop here. Thank you, Caitlin. Thank you, everyone. And if you have any questions, please email me. You have my email below. And I hope that was informative. Thank you.
It looks like a couple comments came in. This one is for Mark. One thing that caused snails to take off was the move away from furrow irrigation that used to drown them. And then in brackets says my dad. Skirting trees sure helps keep them out of trees too.
Great, thank you. Anybody got any comments on copper bands? Is anybody using copper bands or applying copper slurries to trunks? I really would like to check that out and get photographs, see how all that works in the field.
I don't see anything yet to that, Mark, but looks like we do have one question for Ivan. So sugar is listed as 25% of what? Weight or volume of gel liquid?
Yeah, the 25% sugar goes into a water when you're mixing a water and a poison or a pesticide, insecticide, and then you drop the beads inside those that container. So essentially, it's a 25% of the whole gel volume, I would say. But when you're mixing these gels, you're first making the gels and then you're dumping them into a hot tub with the 25% sucrose water with a chemical.
Looks like we have a question for Mark.
Oh, right.
If decalates are not that effective against brown garden snails, they are hard to find in insectaries anyway. Is the best option for organic growers the cultural controls that you mentioned?
Yeah, I think so. I've spent a lot of time looking at decalates in the field, revising the literature. As far as their predatory behavior goes, I don't think they're effective predators against brown garden snail. So if the decalates really are removing leaf litter that the brown garden snail needs, you could speed that process up yourself with some sort of sanitation methods. I know it sounds very impractical to remove the fallen leaves, get rid of the drop fruit, do all the weed control and stuff, but that would remove a lot of resources that brown garden snail can use to, develop and obviously they need it for reproduction. And then that cultural control, at least in my opinion, I have not practiced any of this myself. I mean, maybe, with the skirt pruning, the barriers don't seem to have a lot of use in the field either. So those might be your best options. And if you're going organic, there are some baits that are OMRI approved. So you may be familiar with those baits. I think they're iron-based baits. So between those three things, maybe some sort of ground sanitation, skirting, and maybe OMRI approved baits, you may have the basis of a good brown garden snail management program. The other thing I'd be interested in learning more about, if anybody's on the line, would be, I've seen at least 3 other species of snail in pretty decent numbers in citrus orchards. I haven't followed up on the identity of these snails. I showed some photographs of what might be a milk snail, clustering in high numbers on trunks and eating fruit in San Diego orchards. If anybody's got comments on those 2 snails, and this other one that has a very pointy shell, not a decolate snail, but looks more like a snail that you would find at the beach. I'd be interested in knowing more about that as well, if anybody's got comments or observations to share with that, on that. Thank you.
And then looks like we have one more question for you, Mark, as well. It says, what is the new snail?
Right. Oh, that's just the thing I'm curious about. Okay, so yeah, those photographs I took were pretty bad, and I had a hard time identifying the snails based on doing a photographic search. They're either milk snails, I think, or what was the other one? The white snail, and they're both Mediterranean origin, and probably been accidentally introduced through trade in live plants. So yeah, one thing I think would be interesting to do would be to run pretty much a survey of snails from the San Joaquin Valley right through to San Diego County to see what species are out there and how prevalent they are, and if there's geographic variation in the distribution of these different species. That's something I think would be pretty useful to do. So yeah, just not up to speed on snail identification yet, but I'd like to get there.
And I think we've got time for one more question, Mark. This will also be for you. Are there any effective methods for getting snails out of the canopy?
Gosh, boy, you guys are asking me hard questions. Once they're up in the leaves, gee, I don't know. Back in the old days when I was reading some of Ted Fisher's old literature, they would go in and spray the canopy with insecticides to kill the snails. But I think a lot of those products they used to use are now not registered for use in citrus anymore. So like the carbamate, some of these carbamates, for example, they used to spray. There was a sprayable formulation of metaldehyde too that used to go onto the foliage and could be sprayed on the ground. I think that's been deregistered for use in California as well. So I don't have a good answer on how to remove snails from the canopy. If anybody could answer that, I'd be interested in knowing, what people are doing to mitigate those problems in the canopy.
It doesn't look like there are any more questions. We will stay on for a few more minutes to make sure everyone has a chance to finish the survey. But if you have any questions about the webinar, you can email events@citrusresearch.org. A recording of this webinar will be available on our website, citrusresearch.org, in the coming weeks. I would like to thank Mark and Ivan for sharing this information with us today, and all of our participants for joining the Citrus Research Board for today's webinar. We will stay on for a few more minutes to answer any more questions and make sure everyone can fill out the survey. But other than that, thank you for attending today's webinar.